Hi there folks, my name is Elliott Kimmel and I'm a high school biology teacher at Central Secondary School in London, Ontario, and the course that we are dealing with here is grade 12 biology in the photosynthesis section, and I want to talk to you today about factors affecting photosynthesis. The reference for this is Nelson Biology 12. That's the textbook we use, those pages given to you there, and during the video you can pause or rewind whenever you need to, but you need to take notes. Basically all the text that I write in this video here, draw the diagrams as well, or get them from the textbook. You can scan them, you can photocopy them, or you can draw them from the text. The goal is to come prepared in class to discuss the content of the video, having learned the concepts from the video, and then to be able to go on and look at extensions to this. Chances are there won't be an online quiz for this particular one, but we'll see what happens at the end. All right, here we go. So there are three major factors that affect the rate of photosynthesis, and we've already studied the light and the dark reactions, or the light dependent and the light independent reactions of photosynthesis. So now we want to see what are the factors that affect the process. First one is going to be light intensity, and we'll have a look at that. Carbon dioxide concentration and the temperature. So let's have a look at all three of those and see how they affect the rate of photosynthesis. So the first thing we'll do is we'll look at the photosynthesis equation itself, and you can see that 6 carbon dioxides react here with 6 water molecules in the presence of light energy, and that's going to produce our products, which are carbohydrates, and this is the empirical formula for carbohydrate. If you can imagine a 6 was right there, you'd get 6 carbons, 12 hydrogens, and 6 oxygens. So this could be your glucose molecule, and of course those could be polymerized together to make starch or cellulose or whatever. And 6 oxygen molecules are released or liberated, and those come from the photolysis of water. So here we see a diagram, and we've got light or dark, and we are looking at photosynthesis. There is a process called photorespiration, which we'll have a brief look at in this video, but mostly talk about later on. In photosynthesis, carbon dioxide is coming into the leaf here and being used. Oxygen is being liberated, so we see that as a product over here. Of course, light energy is needed. Photorespiration actually uses oxygen instead of carbon dioxide and actually releases CO2. So this is not a good thing for photosynthesis, where you want to be fixing carbon dioxide. Cellular respiration is also occurring in leaf cells because there are mitochondria, so the carbohydrate that is produced by photosynthesis here can be used in cellular respiration in the presence of oxygen. So oxygen can be coming in here or being produced and used, and CO2 of course is going to be liberated as well. In the dark, cellular respiration is going to dominate. Also notice that cellular respiration is essentially the reverse of the photosynthesis equation. Carbon dioxide is used in, oxygen is used in cellular respiration, combining with some carbohydrates, so oxidizing it and producing water and carbon dioxide. So how are we going to measure the rate of photosynthesis if we want to see what are the factors that affect it? Probably the most common one that you're going to see in the literature is the rate of carbon dioxide uptake. That seems to be the standard. Although in biology labs, students often look at the rate of oxygen release, sort of counting oxygen bubbles for an aquatic plant. And there are others as well. I suppose you could imagine if water is a reactant here, the use of water could be a measure to some extent of the rate of photosynthesis. But we're going to focus on the rate of CO2 uptake. Light intensity and the rate of photosynthesis. All right, let's look at the effect of light intensity and how that causes photosynthesis to be affected. So here we have the irradiance, and this is measured in candelas, or CD. Light intensity or irradiance. We're not going to get into the physics of that, but just imagine you've got a scale here going from 0 and up, so the light intensity is increasing as we go this way. And we're going to be looking at the rate of CO2 uptake, as I mentioned a moment ago. This is called a light response curve, and basically what you want to focus on is this line here. All right, that's the rate of photosynthesis that's going on as it is affected by the light intensity or radiance and sort of measured by how much CO2 is being taken up. So, as the light intensity goes up, you're going to see the rate of CO2 uptake is going up, and that's sort of our measure of photosynthesis, photosynthetic rate. So the light response curve shows the effect of increasing light intensity on the rate of photosynthesis. The dark respiration rate that you can see down here is when the net CO2 uptake is negative, because photosynthesis is, for the most part, not occurring at night. They're not using up the CO2, but they're actually producing CO2 from cellular respiration, which occurs at night. So the actual net uptake is negative because you've got a more positive production of CO2. Where the CO2 uptake, which would be our photosynthesis, and CO2 evolution or release, that would be our cellular respiration, where they're equal, this is called the light compensation point, okay, where those two carbon dioxide levels are equal. As we continue to provide light and photosynthesis occurs and CO2 is being taken up, we eventually get to something called the light saturation point. This is the irradiance level or the light intensity level at which the carbon fixation reactions, the Calvin cycle reactions, where they reach a maximum rate, or where that becomes the limiting factor. And we're going to talk about this light limited area or phase and the CO2 limited area as well. But basically what we see is that as the irradiance increases, the rate of photosynthesis is increasing here as measured by CO2 uptake up to a point which is called the light saturation point, and then it plateaus off, and that is going to depend on not the light anymore, or actually the products of the light reactions, assuming they are plentiful. This will now depend on how much carbon dioxide is available. If there is more carbon dioxide available, then this level will continue to go up to a certain extent, and then as CO2 becomes limited, that will plateau off. All right, so let's talk a little bit more about these two sections, the light limited phase and the CO2 limited phase, the blue and the orange level here. So in the blue region, the light limited phase, the rate of photosynthesis increases, as we've seen, in direct proportion with the increase in irradiance. And this is the light limited phase. So basically what that means is as you continue to shine light on the plant is going to be producing NADPH and ATP, as you know, from the light reactions. And the more light there is, the more energy to kick those electrons out of the pigments and down the ETS, pump hydrogen, and eventually make ATP and NADPH. And this is a diagram showing the light reactions, of course. The photosystems here, and light is coming in right here at both of these photosystems, and the electrons are moving down the ETS. Hydrogen is being pumped into the thylakoid and going back into the stroma through the ATPase, and ATP is being produced and NADPH is being produced, and that is what we need from the light reactions. Now, oxygen, of course, is evolved as well, which we mentioned before. But these are the products of the light reactions, and as irradiance increases or light intensity increases, you make these more and more plentiful. So this region is light limited. As you continue to increase the light, you continue to create those products, NADPH and ATP, and those then are going to feed into the Calvin cycle and be used by the Calvin cycle to produce the carbohydrate. Now, at the light saturation point that we said, though, the NADPH and the ATP can't be used any faster by the Calvin cycle due to the availability of carbon dioxide. So if there's lots of NADPH and ATP and lots of CO2, then you're going to be able to use those products of the light reactions in the dark reactions or the light independent reactions, and everything's fine. But just because you have tons of ATP and NADPH doesn't mean photosynthesis will go on forever, because it's going to depend on how much CO2 you have, because the use of the CO2, as I'll show you here, all right, here's the CO2 right there, carbon dioxide. Well, in order to use this stuff, you need ATP and NADPH, right? So just because you have tons of this stuff doesn't mean that you're going to make a lot of your carbohydrate down here, your photosynthesis product, because you need carbon dioxide. So the light saturation point is the point at which, just because you have these products here doesn't mean photosynthesis is going to continue on forever, because now it depends on how much carbon dioxide you have. If you have lots of CO2, then you can use those products, the ATP and NADPH, you can use them for the Calvin cycle. If you don't have enough CO2, then you could have a gazillion molecules of this and it just wouldn't matter. The whole thing is going to level off based on CO2. So this phase is CO2 limited because the availability of CO2 determines how quickly the ATP and NADPH can be used. All right, now these curves basically show the same thing, what we were talking about. As you can see here in this graph, the irradiance level or the light intensity, as it goes up, so as it moves in this direction, you get more and more intense light. The rate of photosynthesis goes up, however you want to measure that, doesn't matter. So it goes up, but then it plateaus. And what we've got here is we have low CO2 here and high CO2 here. All right, so we're going up here, we're making ATP and NADPH and everything's going great. But at some point, the light saturation point here, we're going to plateau here, and this is the area that is limited by the carbon dioxide availability. If there is more carbon dioxide, then photosynthesis can go to a higher point, right? We can get a greater rate of photosynthesis, but it's still going to level off if there is, a limited amount of CO2. You can only go so high. So in region A, the rate of photosynthesis is increasing with increased irradiance or light intensity and the availability of carbon dioxide. If both are there, you're going to get a nice increase in photosynthesis, and this is really the light limited section. I mean, if you have, sort of lots of CO2, then it depends on how much NADPH and ATP that you can produce, but you got the CO2 to use it. So as NADPH and ATP are made in the light-dependent reactions, they're used up in the light-independent reactions. In region B, however, both curves plateau at the light saturation point, and that's where the availability of CO2 limits any further increase in photosynthesis. And with greater CO2 concentration, as we see here in the red curve, the rate of photosynthesis is higher because any NADPH and ATP that you produce can be efficiently used. But keep in mind that this is going to level off at some point regardless, and I want to talk about that next. So I just drew this curve here. It's the same basic idea, and what we want to say is, if you have a limited amount of CO2 at the light saturation point here, the ATP and NADPH that you produce right here doesn't matter how much you have because you have limited CO2, and therefore the rate of photosynthesis is going to plateau right there. Even if you have unlimited CO2 and you have lots of light intensity, so you make more and more NADPH and ATP, and that's what that represents there, and you got more CO2, so it can use it in the Calvin cycle, at some point it still plateaus. Even if you have 1 trillion CO2 and 1 trillion of these, it plateaus. And this is the concept of enzyme saturation. Even with high light intensity and unlimited CO2 availability, the rate of photosynthesis will still plateau because the enzymes of the Calvin cycle reach their maximum rate and they can't work any faster. So remember, enzymes are finite molecules that have to bump into a substrate in order to convert it into a product. So if you can visualize the Calvin cycle again and you see the CO2 coming in, there's an enzyme called Rubisco there that's going to convert it into its first C3 molecule, and et cetera, et cetera. Those are all enzyme catalyzed reactions. And just because you have tons of NADPH and ATP and tons of CO2, and you say, okay, so work faster, CO2 and combine with this and that, and use the ATP and NADPH, and the enzymes have only 2 hands, let's pretend, right? And they can only work so fast. So even if you have unlimited everything, the enzymes are going to reach a point at which they just cannot work any faster, and this is called enzyme saturation. Think of it sort of like competitive inhibition of enzymes. The fact that a substrate has to bind with the active site of an enzyme. I mean, it's going to take, first of all, time for that to occur. And if you had, say, 1,000,000 enzymes and with 1,000,000 active sites, if you have 2,000,000 substrates, they just can't, the enzymes just can't handle it. There's only 1,000,000 active sites. So the point is that even with unlimited resources, whether it be light intensity, one of our factors, or carbon dioxide, another one of our factors, the rate of photosynthesis will eventually plateau. All right, let's look at the effect of temperature on the rate of photosynthesis. Here we have a graph of temperature in degrees Celsius versus the rate of photosynthesis, again looking at CO2 uptake. And what you can see here is that from about 10 degrees to 30 degrees, we get an increase in the rate of photosynthesis. And beyond that, we get this drop-off of the photosynthetic rate. The light-dependent reactions are not really greatly affected by changes in temperature, but the Calvin cycle is. And that's because the Calvin cycle is catalyzed by enzymes. And enzymes are these molecules, protein molecules, that can work quicker with a slight increase in temperature, but they do require an optimal temperature, again, being protein enzymes, right? Between 10 and 30 then, as the temperature is increased, the rate of photosynthesis increases because these are enzyme-catalyzed reactions. And an increase in thermal energy causes them to work faster, but only up to a point. Beyond that, because they're enzymes, they become denatured, and so they don't work any better. So yes, an increase in temperature can benefit the rate of photosynthesis, but too much is not a good thing. Okay, let's talk about oxygen concentration. Now, this is not one of the things that we originally identified as a factor affecting photosynthesis. We had carbon dioxide, we had light intensity, and we had temperature. But you can see here that in the presence of oxygen, there is a definite effect on the rate of photosynthesis over time. At high oxygen, 40% oxygen here, we have an early leveling off of the rate of photosynthesis versus a lower amount of oxygen. Remember that oxygen is released during photosynthesis by photolysis, the splitting of water. So oxygen is being produced in photosynthesis. Now, the issue is that oxygen and carbon dioxide both compete for the active site on the Rubisco enzyme. This is the enzyme in Calvin cycle. Let me see if I've got the picture here, all right? So here we have a comparison of photorespiration in the Calvin cycle. Recall RUBP is the acceptor molecule that binds normally with carbon dioxide, and we go into the Calvin cycle. Now, there may be some slightly different names here, and it might be looking like it goes counterclockwise instead of clockwise. Don't worry about that. But the enzyme is called Rubisco, RUBP carboxylase oxygenase. And it's called carboxylase because it can take a CO2 gas and fix it onto RUBP, and we get the C3 cycle. Let's see if I can write C3 here, right? And we go on and we make our carbohydrates and everything is cool. Oxygen, however, as it builds up within the plant, if it can't escape, and I'll talk about that in a minute, it starts to compete with CO2 for the enzyme. Now remember, an enzyme is blind. It doesn't have any eyes, right? So an enzyme just grabs whatever fits, like a key going into a lock. Well, if you can picture the structure of carbon dioxide, a linear molecule, you know, like this. I'm not going to be able to draw this very well, but this kind of thing, right? And you compare that to a molecule of oxygen with the double bond like that. They're extremely similar in shape. And so oxygen, as it builds up, starts to bind to this enzyme, and we go this way instead of this way, the way we want to go. And look at this. CO2 is actually released from this process. Instead of fixing CO2, we're releasing it and throwing it away. This is a wasteful process, and it's called photorespiration. It's kind of like respiration because it deals with oxygen. Let me get rid of this diagram here. All right, so we'll talk about photorespiration in class, but basically it reduces the rate of photosynthesis when there's higher oxygen concentration. And this all has to do with not releasing the oxygen in the leaf through the stomates. As it gets hotter, and the stomates start to close, all right, oxygen is produced and it can't escape, and it builds up in concentration, and therefore it starts to reduce photosynthesis. It's called photorespiration. So it's sort of an indirect factor affecting the rate of photosynthesis. So, you know, all this is tied together really. At the lower temperature, say at about 10 degrees Celsius, as you can see here in this blue curve, we get a good rate of photosynthesis, and actually the carbon dioxide uptake is actually greater at first. Plants do better, or C3 plants do better in the lower temperature, because as the temperature gets increased, although we have the increased of movement of molecules, and therefore those enzymes will work faster, the stomata at higher temperature, they start to close to conserve their water. In the end, CO2 can't enter very well or very frequently, and oxygen can't exit, and so you start to get that photorespiration. So what they're saying here is, in this early curve here, early part of the curve, at the lower temperature, the stomates are sort of functioning a little bit better. They're opening great. There's no problem. Oxygen can get out, CO2 can get in, and so we get a good rate of photosynthesis because of those stomates. And here's the stomate. Here's a picture of the bottom of a leaf over here, and you can see the guard cells there, and there's the stoma. So that's where the CO2 would come in. Of course, the oxygen is being produced in the cells, and the oxygen needs to exit. And if you don't open these stomates, the oxygen can't get out. However, we do see that at higher temperatures, we still get an increase in the rate of photosynthesis, because the sort of negatives are outweighed to some extent by the benefit that you get with higher temperature and more enzyme movement and stuff like that. So that a moderate increase in temperature still increases the rate of photosynthesis overall, up to a point, of course, where you're going to get the stomates to close very, very frequently, and therefore you're not going to get good CO2 entry and oxygen exit. So you're going to get a lot of photorespiration, and if the temperature gets too high, you're going to get denaturation. So the C3 plants, the types that we see in the city here of London, most trees and grasses, they tend to do better in cool weather. All right, so those are the main factors that affect photosynthesis. We've got our light intensity, our carbon dioxide concentration, and our temperature. And of course, we see the influence of oxygen, which is related to the temperature too, with this opening and closing of the stomates. What I want you to do is to refer to your textbook. Much of what I've talked about is in the Nelson textbook, so you can read through those pages. In addition, have a look at the section 3.5 questions on page 178. Skim through those. You should be able to answer those questions based on this tutorial if you understand, and you should be able to come prepared to discuss these questions. All right, so I hope that helped. See you next time.